Good morning, everyone. Welcome to the workshop. Today it's my great pleasure to share with you my knowledge on regulated bioanalysis of large molecules. My name is Jinhui. I'm a chemist at Division of Product Quality Research, Office of Testing and Research, Office of Pharmaceutical Quality within CDER, FDA. As always, this is my disclaimer. Here are the learning objectives. First, BPCI Act and the transition of regular pathway for certain biological products. Then, workflow and special consideration for bioanalysis of large molecules in biological matrix. Last but not least, evolution of the bioanalytical methods. Regulated bioanalysis is a quite complex topic. Within the 20 minutes, I can only give you a snapshot of the regulatory requirement, workflow, and current update. First, I'm going to talk about the diversity of large molecular drug entities. Then, in order to put the talk into regulatory context, I'm going to talk about the BPCI Act and the Purple Book. In the main body of my presentation, I'm going to use three case studies: polypeptide approved under NDA, biosimilars approved under BLA, and antibody drug conjugate approved under BLA, to show you what was the bioanalytical work were included in their submission and how they were done according to bioanalytical method validation. Then I'm going to talk about the recent programs in hybrid LBA-LC mass, which is a quite hot topic and more being used. In the end, I'm going to share with you some closing thoughts and two very important reference papers in this field. The diversity of large molecular drug entities will keep increase as more and more new modalities are being discovered and translated into clinical stage. Today, I'm going to only focus on peptide, therapeutic proteins, antibody, and antibody drug conjugate. On March 23, 2010, the Biologics Prize Competition and Innovation Act of 2009, also known as BPCI Act, was signed into the law. It provided an abbreviated license pathway for biological products that are demonstrated to be similar or interchangeable with an FDA-approved biological product. One thing to note is that biological products approved in NDAs that are deemed to be BLAs on March 23, 2020, will be removed from the Orange Book, also known as FDA's approved drug products with therapeutic equivalence evaluations, and added to the Purple Book, known as FDA's list of licensed biological products with reference product exclusivity and biosimilarity or interchangeability evaluations. Right now, you can find the transition biological products in the Purple Book. Here is the link for the Purple Book. Let's talk about the analytical method used for large molecules in biological matrix. Basically, all the assays fall into two categories: ligand binding assays such as RIA, ELISA, and liquid chromatography coupled with mass spectrometry. Back in 1970s, radioimmunoassay, also known as RIA, were used to quantitate hormones and endogenous proteins in human plasma. Later on, multiple detection systems such as ELISA, chemiluminescence, bioluminescence, electrochemiluminescence were developed to reduce the use of radioactive materials. LC-mass was the golden standard for therapeutic drug monitoring of small molecule drugs, but recently it was more and more used for the quantitation of large molecules, and its advantage has been increasingly recognized by both academic and industry settings. It's very hybrid assays, capture mass spectrometry. I'm going to talk about this topic in a later time. Both ligand binding assays and LC mass method should be developed, validated, and implemented according to bioanalytical method validation guidance. FDA bioanalytical method validation guidance was finalized in 2018, and we expect the ICH M10 guidance be finalized late this year or earlier next year. There's a tons of discussion on the details in all the guidance, can be found in all different kind of workshops and reference papers. Bioanalysis of large molecular drugs is not easy. Why? First, structural diversity. You might still remember the slides I just showed on the structural diversity of large molecular therapeutic modalities. Molecular weights range from a couple thousand for polypeptides to 150,000 for monoclonal antibodies. Some of them manufactured by chemical synthesis, and most of them manufactured by recombinant pathways. And some of the large molecular drugs are truly endogenous components, such as growth hormones, and some of them have exogenous components, just like pegylated proteins and antibody-drug conjugate. And even the chemical characterization of large molecular drugs can be very challenging, let alone bioanalysis. Also, typically, you need to deal with multiple analyses in your submission. Let's take antibody as an example. You need to quantitate the antibody in human plasma or serum, but at the meantime, very likely you need to quantitate anti-drug antibody and neutralizing antibodies in the plasma as well. And some of them need the cell-based assays, which can be quite challenging. Also keep in mind the specificity, because lots of the large molecular drugs have endogenous component. In addition, anti-drug antibody, hemolysis, all those factors can cause specificity issue for your analytical method. Stability is also something you need to really keep in mind, because large molecular drugs typically they are liable to chemical degradation, and sometimes even a tiny modification to their structure can lead to the change in biological activities and immunogenicities, and these tiny changes might not be captured by your current analytical method. Last but not least, critical reagents. Not like LC mass-based method for small molecules, which typically only use regular chemical reagents. Large molecules, most of them, they were using ligand binding assays, which require very reliable and robust antibody. Because the structural complexity and the diversity of large molecular drugs, I would like to bring your attention to the concept of fit-for-purpose method development and validation. The term fit-for-purpose for bioanalytical method validation has been a topic of extensive discussion in past decades or even longer. A fit-for-purpose approach is applied when the assay does not fully comply with all current regulatory guidance requirements, but still has scientific and technical validity. As a general principle, the extent of the validation of an assay should be in line with the end use of bioanalytical data generated and should be adequate to support the decision based upon this data. Whether it's a full validation or a fit-for-purpose validation approach, the purpose of an assay is to demonstrate that quantitative measurements generated under specific assay condition will yield accurate and precision determination. Let's move to the case studies, see what the bioanalytical work were included in the application and how they were done. The first case is a recombinant polypeptide. It's an endogenous hormone. I won't tell you the molecular weight, otherwise you can figure out what is it. It was approved last year as a NDA. There are 3 analyses in the application. The polypeptide itself was quantitated by ELISA, which there is a commercial kit available. And the second analysis is antibody-drug antibodies. It was quantitated by ELISA method developed by the sponsor. And the last analysis is a neutralizing antibody, which was quantitated by cell proliferation-based assay, which there's also commercial kit available because this function of the hormone has been extensively studied. The next case is a pegylated growth hormone. It was approved in 2018 as a BLA 351K biosimilar. You can see from the picture, left side is the hormone itself, and the right side is the PEG tail. In the submission, there are 3 methods to quantitate the pegylated protein, anti-drug antibodies, and neutralizing antibodies. For the pegylated protein itself, it was using ELISA method, using biosimilar as standards. The sponsor compared the assay performance using both reference product and the biosimilar as standard. In the end, they decided to use the biosimilar as standard. And for anti-drug antibody, the sponsor developed the ELISA method. And again, they were using cell proliferation-based assays for neutralizing antibodies. For the sake of time, I will not talk too much about the regulatory landscape for anti-drug antibody and neutralizing antibodies. I think our colleagues from OCP will cover some topic in the later presentations. The next case is a monoclonal antibody. It was approved last year as BLA35K biosimilar. The sponsor developed the ELISA method for the antibody itself and electrochemiluminescence method for the anti-drug antibodies. They were using cell-based assay for neutralizing antibodies. Here, I want to bring your attention that for the antibody assay, ELISA method was using the reference product as standards. The sponsor compared the assay performance for using both reference products and biosimilar as standards, and the assay performance were pretty much same. Then they decided to use the reference products as standard. This is something quite unique to BLA products. The last case study is my favorite. It is an antibody drug conjugate approved last year as BLA351A. Please keep in mind it's BLA351A, not 351K biosimilar. It has endogenous component, which is antibody, and also exogenous component, which is a cytotoxic drug. The sponsor developed electrochemiluminescence method for the intact anti-drug antibody and the total antibody, using the intact anti-drug antibody as standard. They also developed the electrochemiluminescence method for the anti-drug antibody, anti-ADC antibody. Since it's an antibody drug and conjugate, the sponsor also had the LC mass method for the free drug, which is the indication of systematic toxicity. A couple minutes ago, I was saying that the assay for bioanalysis of large molecules basically fall into two categories, ligand binding assay and LC mass. After I went through the 3 case studies, you already realize that most of the assays in the 3 case studies were ligand binding assays. Please keep in mind, these case studies had all been approved, so the discovery might already happened a couple years ago or even a decade ago. With the development of LC mass method, LC mass coupled with ligand binding and immunoaffinity capture its advantage has been increasingly recognized by both academic and industry setting. Let's compare the ligand binding assays versus LC mass. For ligand binding assay, you need a specific antibody, which really requires time to generate. But for LC mass, it's pretty much straightforward. Ligand binding assay can have very, very high sensitivity. And fortunately, LC mass, the sensitivity is relatively low compared to ligand binding. Talking about the specificity, ligand binding assay and LC mass, they are specific in different way. But generally speaking, LC mass has higher specificity. It can detect subtle change on a large molecule. As I mentioned, LC mass, the method development is quite straightforward, so it's easy to be multiplexed. But for ligand binding assay, if you want to have multiplexing assay, you need multiple specific antibodies, and you need to make sure you don't encounter any specificity issue. In the end, ligand binding assay typically can be quite high throughput, but LC mass method, especially for the sample processing, can be time consuming and need a complicated workflow. In the next few slides, I'm going to show you the discussion on the comparison of 2 methods among scientific community and how to make decision when to use ligand binding assay and when to using LC mass-based assays. This slide shows the white paper in bioanalysis in the past 10 years. You can see there's always a hot topic to compare ligand binding assays versus LC mass. No later than 2010, people started to propose using immunoaffinity capture to increase the specificity and sensitivity of LC-mass for large molecules. And there were LC-mass for bioanalysis of pegylated protein drugs reported on 2011 white paper. 2013, the white paper title is Hybrid, the best of LBA and LC-mass. That's how the terminology of hybrid LBA LC-mass came from. And from 2014 to 2017, the always hybrid LBA LC-mass in the title of the white paper. 2018, there were reported on hybrid LBA LC-mass for both PK, PD, and antibody drug assays. And in the past 2 years, there's consistently increased report on innovation approach in hybrid LBA LC-mass assays. This is the workflow for hybrid LBA LC-mass assay using monoclonal antibody as an example. Basically, the sample were cleaned up and the analyzed were enriched using an affinity capture approach. Then the monoclonal antibody can be quantitated using LC-mass using 3 different approaches. On the right side is a traditional approach, bottom-up. Basically, the antibody was quantitated based on the quantitation of surrogate peptides. And on the left side, top-down approach, which quantitated the antibody as an intact molecule, was developed a couple years ago. It can give you a big picture of the whole antibody with inside the biological matrix. And the middle down was the latest developed approach to balance the workflow and sensitivity. You can find more detailed information in this very nice review paper. Okay, how to make the decision, ligand binding or LC-mass? And if using LC-mass, how to do it? The first question you need to answer was to measure. If you're working with a endogenous hormone or growth factors, which has been developed many years ago, there's a valuable ligand binding method available, then that's the to-go method. But if you're working on a new modality, which you cannot afford time to develop antibodies, or you have specific issue when you perform any kind of ligand binding assays, or if you're working on an antibody drug conjugate, you want to differentiate antibody drug conjugate with payload conjugate and payload not conjugate. Probably you need to think about LC-mass. Second, whether to use immunoaffinity capture. Traditionally, people adopt the concept of immunoaffinity capture before LC-mass to clean up the sample and increase sensitivity. But recently, there's a very extensive discussion on using multiple dimension LC to reduce the complexity to increase the performance of LC-mass method. And second, bottom-up, top-down, or middle-down. Typically, bottom-up is the most sensitive method, but workflow is quite complex. And the surrogate peptides, you might not be able to find a ideal surrogate peptides. And top-down, typically the workflow is more straightforward. However, and also it can give you the big picture of the in vivo pharmacokinetic behavior of the therapeutic protein, but sensitivity is relatively lower than bottom-up approaches. Next question, how to choose internal standard. This can be a talk itself. Because of the multiple charge status of the large molecules, you need to be quite careful when choosing internal standard. And last but not least, data acquisition and processing algorithms. Again, it's a quite complex topic. This 20 minutes can only give you a snapshot. Here are some closing thoughts. First, bioanalysis is a core member of integrated drug development team. You need to keep in mind which kind of bioanalysis method you are going to use as the initial stage of the discovery and development. Second, one size does not fit all. As I mentioned in all those case studies, bioanalysis of large molecules are quite complex. And last but not least, each assay may review part of puzzle, get a whole picture by putting them together. Again, this is a very complex topic. Many people helped me during my preparation for the slides. especially Yu Min from Office of Clinical Pharmacology, Hao Heng from Office of Biotechnology Product, Wen Ying from Janssen, and Fei from Merck. Thank you all.